Welcome to Spring LIB.
Hello students, in this question we are being given an image which shows the STR analysis from two suspects who left their DNA behind at a crime scene. Now the question asked is, explain the terms locus or loci, allele, heterozygous, homozygous and genotype while incorporating information from this chart. The locus or loci refers to a specific location on the chromosome where a particular gene or genetic marker is found. And in the analysis chart, which is STR analysis chart, the loci are represented by rows, which are here, rows labeled D3S 1358, VWA, and FGA, and so on. These are the specific locations on the chromosomes where the DNA samples are analyzed for short tandem repeats for these suspects in this case, which are known as STR. Number second term here is the allele. As we know that allele represents the variant form of DNA that occurs at a specific locus on a chromosome, and in the STR analysis chart, the alleles are represented by the numbers in the columns under each suspect. We can see that here under the suspect or under each loci, here are represented the numbers for both the suspects, which is contributor 1 and contributor 2. As we can see that for contributor 1, the alleles represented are 17, 18, 16, 17, 18, 20, 14, or another one it is 14, 16, 17, 23, 25, and these all represent the alleles or variants of the DNA on a specific site on the chromosome. And these numbers represent the number of repeats of a specific DNA sequence at that locus. Number third term, which is defined in terms of STR analysis, is heterozygous. As we know that an individual is heterozygous at a particular locus if they have two different alleles for that gene. In the STR analysis chart, as we see that the suspect 1, as we can see that the contributor 1 or suspect 1 is heterozygous for all the three alleles, all the three loci, this, and this one. As we can see for each loci for the suspect 1, there are two different alleles, 17 and 18, 16 and 17, 18 and 20, which means that this suspect 1 is heterozygous for all the three loci. While coming to the suspect 2, the suspect 2 is heterozygous for the two loci, which is B3S 1358 locus and FGA locus only, because here the suspect 2 has 14 and 16 alleles and the suspect 2 under FGA has 43 and 25 alleles, two different alleles, so it is heterozygous for these two loci. Now coming to the another term, homozygous, as we know that homozygous is an individual at a particular locus if it has two identical allele for that gene. As we can see that the suspect 2 or contributor 2 is homozygous for at the locus VWA, which has only 17 allele at this locus. So for this locus, suspect 2 is a homozygous. Coming to the another term, genotype, as we know that genotype refers to specific combination of allele that an individual has at a particular locus or set of loci. In the STR analysis chart, the genotype of the suspects are represented by the combinations of alleles at each locus. For example, the suspect 1 genotype at D3S1358 locus is 17 and 18. So for suspect 1 at D, I will represent this first with D locus, it has alleles, for example 17 and 18, and for suspect 2 under the locus D, it has 14 and 16 allele at the same locus as that of the suspect 1. So the entire set of genotypes across all the loci, which is 3 loci here. So the genotype under all the 3 loci of these two suspects represents the suspect's overall genetic profile. I hope you found the video useful. Thank you.